Good afternoon, everyone. Welcome to this week's Institute for Quantitative Biomedicine seminar. It's an honor for me to be able to introduce today's speaker and to make some additional announcements. Welcome to everyone here in the room and those of you from across the United States and around the world. It's a pleasure for me to be hosting today's speaker, Professor Jeff Gray from Johns Hopkins University. But before I do introduce Jeff, I wanted just to remind you that on the 13th of December, a former member of the Institute for Quantitative Biomedicine, Dr. Anders Laursen, will be here talking about the work that Renew CO2 did. Excuse me? Microphone not working and not standing close enough to it. Okay, that probably explains it. So in addition to Jeff's seminar today, I'll be on the 13th of December, I'll be introducing Dr. Anders Laursen from Renew CO2 Inc. Renew CO2 is a Rutgers startup that was a tenant here in the building before it hit the big time, attracted a lot of venture capital money and also non-dilutive capital from the Gates Foundation, non-dilutive capital being absolutely key to the success of a small company. And he'll be talking about their efforts in trying to take carbon dioxide out of the air and turn it into fuel, which you can understand is an absolutely key innovation that needs to happen among all sorts of other changes to the global energy economy to ensure that we don't commit suicide as a species and destroy the planet in the process. I just wanted to reiterate that there are opportunities here at the RCSB Protein Data Bank at Rutgers and at UCSD in terms of work in software engineering and as postdoctoral researchers, as gap year recent graduates, and also undergraduate summer researchers. So please go to our jobs page, which is www.rcsb.org/pages/jobs to learn more about these opportunities. Thank you very much, Michael. So without further ado, let me just find my script and introduce Professor Jeffrey Gray, who is a full professor at Johns Hopkins University. He was trained at another Big Ten school, the University of Michigan, in chemical engineering, MGO Blue. And then did his PhD in chemical engineering at UT Austin, another big football school. So he was not very impressed by Rutgers football, I have to confess. But that is something that we don't have control over here at the Protein Data Bank and IQB. So Jeff and his team research computational protein structure prediction and design, particularly protein docking, therapeutic antibodies, and glycoprotein using deep learning methods. The Gray Lab leads development of Rosetta Dock, Rosetta Antibody, Rosetta Carbohydrate, the ROSI web server, and the Pi-Rosetta interactive platform for protein structure prediction and design. These tools, as I think many of you appreciate, are literally used by thousands of researchers, both in academia and in industry around the world. And Jeff's team has been very successful in Capri and in other assessments of antibody modeling. His work has been funded by NIH, NSF, DARPA, ACS, the Beckman Foundation, the Rosetta Commons, and two industrial funders, UCB, and the vaccine division of GSK. So, Jeff, it's wonderful to have you here and to learn more about your program and the opportunities for intersection with the Protein Data Bank. Thank you very much.
Sure. Let's turn that up. Can you hear me in the back? Can you hear me? There we go. Thank you, Stephen, for that introduction, and thank you for the invitation here. Football's okay. I'm at Johns Hopkins, so we Division III. It's nice to be at a division school, one school again. Yeah, it's a real pleasure to be here at the home of the RCSB, the Protein Data Bank. I not exaggerating to say my career would not exist as it is without the protein data bank. At least I'd be doing something completely, totally different. It's been so instrumental to have this database of structures to work with and think about. Oh, right. You got me?
Yep.
Okay. Yeah, I've already enjoyed starting to meet people here. I'm looking forward to meeting more people to hear how this operation runs. So I'm going to talk today about artificial intelligence tools for antibody engineering and protein docking. What I'm hoping you get out of this is a little bit of an idea of how these computational methods work and what we might be able to do with them. I've been working on antibody engineering and protein docking for a long time, so I'll talk about both of those particular thrusts in the talk separately, and then we've been doing it with physics for a long time, and this AI thing, as it in the world, it's kind of blasted on the scene, and now we've learned a lot of that over the last few years. So yeah, a couple of years ago, I would have had this talk all be about Rosetta that you've seen in Sagar's group and with work of people here, but I thought I'd shift it up today and tell you about how some of these new methods are working. Okay, so I'm sure this community has seen this slide a few times. This is the performance in CASP year to year with the line showing every 2 years how the performance has gotten better from easy targets to difficult targets, from not predicted correctly to correctly. And AlphaFold 1 was big. And then in 2020, everybody except for AlphaFold. Yeah, so this is Rosetta, I think, pushing things forward for many years. And then we kind of hit a limit, and then AlphaFold 1 makes a jump. And then 2 years later, everybody except AlphaFold has already beaten AlphaFold 1. And AlphaFold 2, of course, gets almost everything quite right. And that's down to even structures of the loops, and the structures look so wonderful. And you have a confidence measure to know if the structures are expected to be correct or not. So this had, all kinds of existential questions for me and for my lab and for all of us in the field about what are we doing and what should we be doing next. So I want to back up a little about what we were doing. We were using physics. And so with physics, this paradigm is that we start with physical laws. And from those physical laws, we put them together and we build models. And from those models, we calculate or simulate or do a search and come up with something about the physical phenomenon in the world. So we start with these underlying laws or ideas or physical ideas, and then predict these behaviors. And then at the end, we look at the experimental data, which in our case was the protein data bank. So I would put in, energies between proteins and things about what's the lowest free energy state. And then I'd try to predict structures of docked complexes. And then I'd compare with the PDB, which I could, without that, I couldn't do anything. And then we'd say, based on that comparison, should we go back and change things? Now, I'm not going to change the definition of Gibbs free energy, but maybe that Coulomb's law, how does it apply for molecules in solution? And what's a dielectric constant either? Does this thing even work? And so we kind of go through this cycle to make better physical models. Of course, now we're in the machine learning era, and the paradigm here is that we start with a pile of data. So that would be the protein data bank, right? And then we split that in the supervised learning case into inputs, which the machine learning people call features, and outputs, which they call labels. And you're going to try and predict these labels from the features by going through some neural network. And so you can go forward through this neural network and see how you do. And then based on the errors, you can go backwards and adjust the parameters in the network. And so you go back and forth many times. When you do that, you're going to minimize this risk, which would be some kind of error function between the prediction, the real labels over some data set. And then you'd minimize that by changing the parameters of the model, these thetas, which are in these different layers of these neural network calculations. So my lab has, over the last 20 some years, worked in a lot of areas. The centerpiece is protein docking. How do proteins interact? What is the interface like? If I'm given two components, can I find how they bind? And there's all kinds of problems that come out of that, specificity or affinity, And then lots of collaborations with different people, but I'm fundamentally a methods person interested in how to make the methods to do these calculations. And we've done a lot of work with antibodies where protein-protein interactions are the fundamental thing they do is recognition. And we've worked on membrane proteins, we've worked on glycoproteins, we've done some with proteins and inorganic solid surfaces. All of that with Rosetta and building the toolkit of Rosetta to handle these different situations, search different configurations, search different sequences, come up with different energy functions to try and get these problems to be able to predict them correctly. And then we always dreamed of the day when we'd be able to do this really reliably. And now AlphaFold has gotten us so much closer to that goal. And then in 2019, I had a summer intern. So this is between AlphaFold 1 and AlphaFold 2. I had a summer intern who was a computer scientist and said, well, let's just go ahead and let's make a convolutional neural net on some images and see what we can do with antibodies. And by the end of the summer, he could match what Rosetta could do that we've been developing for 15 years. So we've kind of since then been switching my lab over. And over the last 4 years, we've tried lots of different things, convolutional nets, multi-track networks, language models, graph models and voxel models and more graph models and then diffusion models are the latest hot thing. So it's kind of been a whirlwind. We've completely shifted what we've been thinking about over the last few years, which has been a lot of fun and sometimes a little terrifying as you're sometimes competing with lots of postdocs at DeepMind and things like that. For today, I thought I'd take you through 3 stories: the multi-track model for predicting antibodies, the language models for designing antibodies and looking at antibody sequences, and then the diffusion model for docking. So that's what we'll focus on. The overall questions that I'm trying to get at in my work, are these AI methods good enough for protein engineering problems? So they could do CASP really well, and that's great, but does that work on practical problems? I'm an engineer, and so can we do useful things with that? How and why do these methods work? And based on that, can I change things? Can I address limitations? Can we trust them? And then how are these methods different from physics? So we have this long tradition of working with physics. Is it the same? Is it different? Can they complement each other? Are they, I don't know. What's the difference? So I hope some of these questions I'll give some insight to as I go through my talk. Okay, so before I get too much further, I do have some conflicts of interest. I'm the director of the Rosetta Commons. We license Rosetta to companies. We give it away free, like the Protein Data Bank, but we license it to companies, and that money comes back in and supports lots of things at Hopkins, including like our intern program and some of our diversity efforts. Rosetta had a spinoff called Cyrus that has a GUI that houses Rosetta, and so and I was on the scientific advisory board, so I'm not anymore, but I have some financial interest there. And then these latest deep learning methods, we haven't really known what to do with them. We explored different IP models. Some of the students have been interested in spinning off companies, so we ended up patenting one of the algorithms, and one of the algorithms we're licensing through Hopkins. So those are the conflicts I have. Okay, so part 1, antibodies. What are we doing with antibodies? Trying to remember if Joey Lubin was a co-author on this paper or not. This is around the time when Joey was in my lab. This is what we traditionally did with antibodies is we wanted to take an antibody sequence and build a homology model, the 3-dimensional structure of the antibody, which itself is interesting because you see the structure. Well, okay, antibody structures maybe aren't that interesting because they all look the same, right? It's just an IG fold. But what's interesting is that paratope where these loops are always different. And the very subtleties in those loops are going to determine how they're going to bind to an antigen or which antigen they bind to. And so my core problem of protein-protein docking, I need to get this very right to be able to do the docking correctly. So we're going to do homology modeling of the antibody and then docking. That's most of what we did for the first 20 years of my career because protein, I'm a computational person. I don't have a wet lab. I can do this computationally because I've got the protein data bank to tell me if I'm right or wrong. I can develop methods and then see how right or wrong I am. Today, we're also trying to push this to the next step. If we can do these pieces, maybe we could do design. So we've been doing a lot of efforts in trying to design antibodies to bind things. The holy grail problem being, here's a new target for cancer or an infectious disease. You need to bind this patch to be effective and come up with an antibody or 10 antibodies, and most of them actually work and bind and are soluble and get through the lab. That's kind of the driving vision on the antibody side of the lab. Okay, so an example of this, this was a postdoc in the lab, and actually she came over to us when she was a PhD student in Inger Sandley's lab. Rahel wanted to know about celiac disease, which is, of course, an autoimmune disease where you eat gluten and there's some kind of allergic reaction to gluten. The gluten gets broken down into gliadin. Gliadin gets presented on MHC molecules, and then that's somehow creating this allergic or immune overactivity. So what Rahel was doing was trying to raise antibodies that bind the peptide MHC complex. She made some of those with selection techniques in the lab. In her home lab, she came to Hopkins, built three-dimensional models of the antibodies and docked them to these peptide MHC models, came up with structural hypotheses, so how this recognition worked, and then thought about, could I extend a loop or change something to alter the specificity or make it bind stronger? So this turned out to be very successful. We ended up coming up with models that her experiments had a lot of validity to them, and she was able to increase the affinity of the binding, and then they were able to pull out which cells with an antibody that was binding strongly, they could bind strongly to the cells that were presenting these, and they were able to determine which kind of cells were presenting this antigen, which is a big part of how the disease works. All right, so that's the idea of what we want to do and how we want to do that. Okay. Flicker. Okay, there we go. Okay, so today, why don't I just use AlphaFold for this? AlphaFold's amazing. It does a lot of good stuff, but one Achilles heel is antibodies. And there's a couple reasons. AlphaFold relies heavily on the multiple sequence alignment, the MSA. They're going to line lots of sequences up and find this residue here and this residue here, mutate at the same time, so maybe they're close in structure. So your antibodies evolve in an individual organism, though, over 7 to 10 days while you're fighting an infection. So the antibodies in the public databases are not necessarily relevant. You'd really want the antibody sequences in the repertoire of the animal that's mounting the immune response. And to get the docking, you really want the mutations correlated with the antigen. But the antigen, the virus, is changing over timescales of years, right? So the virus changes and then the antibody. So you don't see these correlated mutations. So AlphaFold's biggest information source of these multiple sequence alignment is pretty useless with the antibodies. Yeah, and so here's some docking. This is from CASP 2022, the last CASP 15, I guess. Here's the experimental structure, and here's the structures that came out of AlphaFold 2 multimer, and you can see they're all just bound to the wrong place. So we need to do better. Okay, so let's back up. Let's do just first the homology modeling problem. Can I use deep learning to predict the high resolution structures of antibodies? That's the first thing we worked on because my lab's been doing that a long time, and we wanted to see how these other methods did in comparison to Rosetta methods. This is the intern that got us all started. Jeff Ruffalo took it over and had the intern's paper by the end of his rotation. And then this is another intern, and I didn't know anything about deep learning, so I have a BME professor who is an expert in deep learning who helped guide us through as we were learning these pieces. So I mentioned this is the intern's problem. We used a convolutional neural net, which was used to recognize images. And so we had to make a protein look like an image, which you do by doing contact maps or distance maps. This is residue 1 to n and 1 to n and how much distance between them. And then other geometrical quantities, which we took from the TR Rosetta approach out of David Baker's lab about the angles. There's actually 6 different numbers that completely defines the translation and rotation between 2 amino acids interacting to each other. And I really like this conceptual idea because it's like physics, right? These 2 amino acids are interacting energetically and hydrogen bonding or varying some hydrophobic surface area. So by defining those 6 dimensions, maybe it's going to pull out something like the pairwise interactions between amino acids. So we use these convolutional nets, and I won't go into this story a long time because these are a few models. They're all of 3 years old, so they're like ancient in terms of deep learning technology, right? We took the sequence, we used a convolutional net, we predicted these, then we stuck it into Rosetta and we found structures. That was called Deep H3. We first started just predicting the H3 loop of the antibody, which is the hypervariable difficult loop. Then we added side chains. This was a different summer intern who added side chains to see if they would be more accurate. And then we also made a model that did the full variable region of the antibody and also kind of identified which regions were important in making that prediction. So one of the nice things about these deep learning methods, unlike physics where I just say, hey, it looks like a great energy, it should be good, but a lot of times it's not. These you can train the model to predict something, and then you can train another model to recognize when the model is correct or not. So you get this confidence measure out, and we put that into our antibody structure predictor. And what I'm showing here on the bottom is a combination of our confidence in our model, where negative is better to the left. And then this is a data set of antibody mutations, and this is binding enrichment for thousands of different mutants. This is from Sorel Fleischman's lab. And Sorel had made lots of different mutants and then looked at which ones bound or not. So this was data we already had, but we didn't train on. And it turns out if we looked at our confidence metrics and we took things that were more confident, mutations that made the model more confident in the structure, they were highly enriched in things that actually made the binding tighter also, which was kind of interesting because we didn't train this model at all on binding. This model is just about predicting the structure. Nothing about the antigen at all, but by making the structure more confident, so you're maybe positioning that paratope more carefully, it's picking out things that bind more. So retrospectively, all of our top-ranked mutants improved binding affinity, and then this is all retrospective. We started a prospective study with GlaxoSmithKline, and those data are like a year old, and we're this close to finally having the company approval to share them. So I don't have a slide yet, but this has worked in some prospective cases also. Okay, so those are the old models. So the new models, language models, attention-based models, multi-track models, and invariant equivariant formulations, and I'll explain what these things are. So the attention-based model, this is the piece that's at the core of all the deep learning algorithms. I don't know, how many people are programmers and like write and use deep learning algorithms, and how many people are structural biologists? Who's everybody else then? Chemists and other things. Okay. I don't want to get too deep in the computer science, but I like to try and break this down a little bit for those that haven't looked at all the machine learning, so you kind of know where it's coming from. So these network models, they're flow models where information or data flows through layer and layer of the calculation. Just like in our brain, we have input from our eyes, and then things flow through and then come output through our mouth or whatever. And so depending on how you flow this information through, that's what's important. And this is this key structure, this key equation they have is Q, K, and V, queries, keys, and values is what goes into this attention calculation. And there's a matrix thing. And it's basically some matrix multiplications of these three pieces coming together, and then you're training these parameters. We're going to train parameters for Q and K and V. That's what it's going to learn about how to mix these three things together. So let me show you an example of what this means. Here's what a language model is trying to do. A language model, you're taking each word and you want to figure out what the meaning of that word is. So you're going to take the sentence in and you're going to turn them into numbers, and then you're going to bring it through several layers and put it into a really high dimensional space where you have different words in different places to be the meaning of that word. And so you take a sentence like this and the machine has to figure out what does it mean in this sentence, and it will be different depending on different sentences. The cat drank the milk because it was hungry, or the cat drank the milk because it was sweet. The it refers to a completely different noun, right? And how does it know that? Well, what you do, so I'm trying to figure out the value of each of these words, what the meaning of that word is in some sense. And I'm going to do that by looking at how the other words resonate, which could be words that are far away from each other in the sentence. So here, the cat and hungry go together. Those clearly link together. So if I take the sentence, these K, Q, and V's, I'm taking those matrices and I'm multiplying it through by the sentence. So I'm looking at each one of these words is in Q and each one of these words is in K, and I put them together and I'm looking at the pairwise interaction. And it should pick up here that cat and hungry really go together. And in this sentence, it should pick up that milk and sweet really go together. And it needs to learn that. So there's the query and the key. and it's going to punch those together. And then depending on how those pairwise interactions are, then I'm going to project it down to the values of the original thing. So I'm going to figure out what it means based on the pairwise interactions of all of the other pairs. Are you with me? I hope that wasn't too much math. So this is the innovation. This paper is, I don't know, 5 or 7 years old now, but this is kind of underlies all of these, a lot of these breakthrough deep learning methods is looking over the whole piece of chunk of data that you're putting in and figure out what relates to each other and what the model should pay attention to. So you can focus in on things that are important. Okay, so this is a figure from AlphaFold, which heavily uses attention, and you don't have to get all of this, but what I'm showing here is there's these different streams of information of data coming through from the left to the right, and they have multiple layers of these. And AlphaFold has multiple tracks. So this is a one-dimensional track of the sequence. This is a two-dimensional track. And the one-dimensional track has that multiple sequence alignment in it. What's going on in the multiple sequence alignment? The pair representation has how different residues interact, just like I showed in the convolutional model earlier. And then they have a three-dimensional model where they're putting three-dimensional amino acids together in space. And what all these lines that cross over back and forth, those are all attention pieces. And it's probably too small to see, but here's keys and queries, and then there's values, and then here's values and weights. And so all of these crossovers are the one-dimensional paying attention to the two-dimensional and the three-dimensional paying attention to the one-dimensional and back and forth. So it's learning how to pay attention to the 1D, 2D, and 3D data to figure out what each amino acid means and where it should be in space and what it should do. All right, so I hope that it's a bit hand-wavy, but I think that's kind of what's going on under the hood. Let me show you an example from our work, going back to just the language stuff. This is our language model for antibodies, which we call Antibody. BERT is one of the standard language architectures. How it works is we take an antibody sequence, just like you'd have those sentences I was showing you. This is how you train the language ones too. You take the sequence and you mask one of the residues. You hide its identity and you put it through all these layers and you ask it to predict what residue should be there. And so you say for each of those 20 amino acids, what's the probability of that amino acid going there? And then we go, when we're training this model, we take many, many antibody sequences and we go back and forth and fix the parameters in Antibody and have it learn how to make this prediction, how to fill in missing residues. Just like in an English sentence, if you don't hear a particular word, if it's missing or dropped out, as you're trying to struggle and hear on Zoom, then your brain might fill in what word might go there. You kind of guess what goes there. Okay, so what's fun about this is we train based on sequences, but things in structure pop out right away. So one thing people observed is that in protein languages models, secondary structure pops out in one of the outputs that comes out. We found that disulfides in the antibody structures, those are something that pops out. It knows that those two residues belong together. Okay, so how do we train this? There's another great database called the Observed Antibody Space out of Charlotte Dean's lab. At the time, they had 1 billion sequences of antibodies there. Now I think there's like 3 billion, and most of them are COVID patient antibodies. We took a somewhat non-redundant set. You have to keep a lot of the redundancies there because antibodies are redundant. They come from the same chunks of genes. But we ended up with 0.6 million, or about 600 million sequences. We trained on those and validated on it. The model has 26 million parameters in it, so it's a really big fitting function. So you put the sequence in, you can mask things, you go through all these layers, and then at the end you predict the probabilities. But before you predict the probabilities, the last layer, each node for each residue has, say, 1,000-dimensional vector that represents the meaning of that amino acid. And that we call the embeddings, or the final hidden representation of what the model has learned about that position. You can take those embeddings and see what kind of information is in here. You can take those embeddings and plot them on what's called a t-SNE plot, which is kind of like a principal component analysis. It just spreads out the data. And then here we're coloring it by the canonical classes of the H1, H2 loops, which fall into canonical groups. And you can see they pop out neatly, the different colors of the different clusters. So that this clustering of different loops came out naturally from the language model, even though there's no structure taught to the model. And H3, which is hypervariable, doesn't have that clustering, although there's some pattern here with the length of the H3 loop. Okay, so that's the That's the sequence-based model, and you could use that if you didn't know an amino acid, you could plug it in. That might be useful for design. We were interested in structure. I didn't want to use multiple sequence alignments like AlphaFold does. So this is a way to put in information about the sequence that's not a multiple sequence alignment. So we take the antibody sequences, we feed them through Antibody and get these sequence embeddings, and we feed it to this new model that we have to predict structures of antibodies. It's going to predict a backbone, and then we'll use Rosetta to put the side chains on. IG-fold is that model. It's a fully connected residue graph with 3D coordinates inside it. We borrowed heavily from AlphaFold for this. We took components from AlphaFold and kind of adapted it for the antibody data set. We train it to minimize the loss of the structure prediction. So we want to get the bond lengths and all the structure correct and get the coordinates in the right place. Okay, so this is the full inside of that IG-fold model. We feed in Antibody, we get these sequence embeddings, we take a vector product of those two to get a 2D track, and then we have a 1D track and a 2D track. We put it into graphs. Triangle updates are from AlphaFold that have another way of getting 3-body interactions, and then we have this 3-dimensional template structure we put in, and then this IPA module is that picture of AlphaFold I showed you before that has the multiple tracks that all talk to each other. We spit out antibody structures and then separately retrain a module that can predict how much error is in the antibody structures. Okay, so there's only about less than 10,000 antibody structures in the protein data bank, only 4,000 unique ones that are useful for training on. That's not a lot to train a model. AlphaFold used many more structures. So what we did is we took the actual structures of antibodies out of SABDAB, which is a sub-database feeding from PDB. And then we took sequences that were known of antibodies, and we predicted structures with AlphaFold. So we used AlphaFold to make structures, and we used the AlphaFold structures to train things. As somebody trained in physics, that seems crazy. You put garbage in, you get garbage out. If you have nonsense, your model's going to learn nonsense. I'm still getting my head around this. My neighbor does computational linguistics, like machine translation, and he tells me, no, in machine learning, that's totally wrong. In machine learning, you want your model to have lots of data, even if it's bad data. You start with bad data and you get better. And I've shifted my thinking a little bit. It's my, I teach as a professor, I do research, but when I teach, the machine learning is almost like teaching. I'm teaching this thing to do it. When I teach my kid to talk, if my kid talks in baby talk, that's okay, that's good. It's making mistakes and learning as they go. And it's the same thing here. It's okay to get some nonsense and some garbage in a little while as you're learning and getting better. So what we do is and so this is done machine translation all the time. If you have a language where there's just not many translated books, you have it practice on those and then have it start training other books. And even though it's translating poorly, your model gets better by learning off those experiences. So, okay, so we put in these structures from AlphaFold, but we're also careful. We made a lot of them, but we make sure we're always anchoring it with the actual crystal structures that are ground truth. So we don't let these take over too much. All right, so all right, there's some losses. How does it do? This is a comparison slide in our paper because by the time scales on these are crazy. We preprint, and you're the only program here, but by the time the paper is actually published, there's like 4 other methods. So these are all the deep learning methods. These are different components, different loops of the antibody and the RMSD loss. So you want that to be low, and the H3 is the hardest. And you can see it's harder than the other ones. We're at about 2.5, 3 angstroms of error. If I put, going back to the classical methods, the physics-based methods, those error are more in the 3 to 4 angstrom range. So there's a definite improvement of all the machine learning models over the physics-based models. The other thing is that our model is so much faster. So when we did Rosetta to predict an antibody, I'm firing up a cluster of CPUs and burning about 300 CPU hours. The machine learning model takes a long time to train, but once it's trained, it's just y equals f of x, and it comes out in a matter of seconds. It depends on some of the details. Of course, AlphaFold needs that multiple sequence alignment, so it takes a lot more time. And then some of the other methods, ours is a lot faster than all of the other methods. So what do we get out of it, and how's that compare to AlphaFold? These are a bunch of predictions, and on this axis is the error that AlphaFold has, and here's the error for IgFold. And you can see there's points on both sides of the line. Sometimes IgFold is better, sometimes AlphaFold is better. And here's two examples. Here's one where IgFold is wrong. Here's one where AlphaFold is wrong. So the models have learned slightly different things. By us focusing on antibody structures, we've learned things that work better in some cases, and AlphaFold, which has learned proteins in general and loops in general, does better in other places. So when we truly don't know, say for the Capri challenges that are just started this week, we'll run both. And we'll look at the confidence measures and we'll see which model thinks it's more accurate. Okay, so here's the confidence metric. Here's the actual error, and here's the predicted error, and it's pretty linear. So the predicting the error works pretty well. You can see there's a weird factor of 2. Our model always thinks it's more accurate than it is. I think this is because it's very accurate on most of the protein, and then the H3 is where it's inaccurate. This is H3-RMSD, so it underestimates that because the rest of the protein's always better. What about nanobodies? So in the review process, people asked about nanobodies, which I didn't want to touch because there's not much data. But it's really important for industry and a lot of drug pipeline. And similarly, sometimes we get IgFold gets nanobodies correct, and sometimes AlphaFold gets it much more correct. Here's one where the nanobody has a nice long H3 because it kind of the nanobodies, the single chain antibodies, they don't have this interface with the light chain, and so sometimes you get these long chains that cover up that heavy light interface. And our model doesn't know so much about that, but AlphaFold, that knows about proteins folding in general, does better at that case. All right, because our method is so fast, we can crank it up and just make a bunch of structures. Ooh, my time's going quickly. So that's what we did. We took this open antibody source and we made 1 million structures. So there's 4,000 in the protein data bank. Now we have 1 million. So that's a lot. They're not all perfect, but we do have them annotated of which ones are better. And so those are potentially useful for training other things and just looking at structural space. Okay, so that's prediction. On the design side, what can we do with design? I want to briefly, actually, I want to get to the diffusion model. So I'm going to go a little bit quickly through this part. This is another summer intern, Richard Schwei, that a PhD student supervised. So in antibody discovery, a lot of times it's done with sequence libraries. So you start with a library of sequences, and then you do a display experiment and see what sticks and keep what sticks, and then put that variable structure onto different backbones to make different formatted antibodies. We were wondering if we could put something computational in the beginning here. Could we make a library of good antibodies that then you could pick things out? When you do this synthetically, if you just make a lot of random changes, a lot of the library isn't functional, doesn't work, doesn't express, is poorly behaved, and so they get downstream, and then all of a sudden it doesn't express well or isn't stable. So what if we could have a generative model that makes a lot of sequences and we put quality control in at that step? Okay, so generating sequences is different than learning representations. So in Antibody, I would mask something and then predict what's there. We're going to change into this generative autoregressive method, which is incidentally what ChatGPT does. If you watch it, it takes what words have been there before and it predicts what's the next word, what's the next word, what's the next word. So we start with a few amino acids, and each time we go through the machine, we just predict what's the next amino acid, and on through. So that'd be great for getting a complete new antibody from N to C terminus. A lot of times, though, I want to be more targeted. I don't want the whole thing new. Maybe I just want to play around with the H2 loop or the L3 loop. So we came up, okay, so here's a little more how this works. We put in some tags. We want a human antibody, we want a heavy chain. It predicts the first residue, and then from the I-1 residue, it predicts the ith residue, on and on till the end. So each time we're getting the probability of a residue given the species of the chain and all the residues that came before. And then, okay, so if we want to do just a loop, we've got to trick these models a little bit. What we're going to do is where that L2 is that I want to replace, I'm going to put a special token in and say, here's a missing piece, something goes here. And then the missing piece I'm going to put at the end. I put a token to say this is the end of the antibody, and then this is the insert piece that goes in the middle. And what's nice about this is that I can get the context of the whole antibody in place and then say, now that you know the whole antibody, can you go back and fill in just loop 2? Does that make sense? So it's a little different than And okay, so that's unique to us. That was an innovation to do this sequence infilling with a generative model. And then here's a case where we take the H3 and we're generating lots of different H3s. We can change the temperature. The temperature at high temperature, you're picking more random things. At low temperature, we're only taking the high probability things. And we can see if we do that and we predict with AlphaFold, what does the structure look like? As I raise the temperature, at some point, things get too crazy and I don't get an antibody anymore. But at low temperatures, it's still an antibody and I can determine how much diversity I want to put in here. Yeah, I think I don't want to get into too much of the detail because I want to show you the diffusion models and have a little time for questions, but we do this for different types of antibodies, different species, heavy, light chain. And so we could ask, if you ask it for a mouse antibody, do you get a mouse antibody back, which we do. When we ask for a rat antibody, not so much because there's not as much data for rats. So that doesn't do as well. Heavy and light chain gets predicted pretty well. And then we played with things like, as we change the temperature, or other internal parameters that change the diversity, how much diversity can we put into the system? And this is kind of the interesting experiment. Then we took therapeutic antibodies. We took 49 antibodies that are in the clinic, and we said, what if we cut out the H3 and then make libraries from each of these? And we can do it really fast because it's in the computer. We made 9,000 sequences, 9,000 new antibodies deriving it from older antibodies like trastuzumab, et cetera. And we predicted structures. And then what do you get? What comes out of this? Well, first, you get different length H3s. You could take out a length 12 H3 and it'll give you different length H3s, which is an advantage to our method. If we did that masked language model like Antibody, we'd always have the same length because it's not changing the length. By generating, we're getting alternate length loops, just like nature does. We can play with the diversity. And then this is the interesting part. Okay, this is all in the computer, so take it with a grain of salt. But in the computer, you can say, is the sequence going to aggregate? Will it be soluble or not? So this is a predictor of aggregation. This is the wild type sequence. And then you can see most of these points have a lower aggregation propensity. So we're generating things that seem to be better behaved in solution. Similarly, that's aggregation. This is solubility. This is both of them. We can look at that distribution in silico, and if we're going to list out the antibodies to put in a library, we can pick the best ones out of this. All right, and then this one would be one thing you look at is the small fragments. Are they human-like? Because that tells you something about maybe it'll cause an immune reaction or not. So we have a proxy for immunogenicity. So we've been working with different partners about trying to get some experimental tests of would this library perform better than random libraries. Okay. Let me skip. That's a big structure about antibodies. You saw structure prediction and you saw design. Docking is the other thing we do. I want to switch to docking for the last 10 minutes of the talk. So this classical docking problem is given to structures. How do they fit together? Lately, we've been doing a harder problem, which is given to sequences, predict the structure. That's what you can do with AlphaFold. Just put the two sequences in and see what gets spit out. But traditionally, we look at two structures. And I'm going back to that problem because that's an interesting problem to me because you can get at binding-induced conformational change, which is still an unsolved problem. When things change upon binding, it's hard to get the right answer. And in the blind Capri challenges, most people get those wrong. Actually, everybody gets it wrong if it's over 2 angstroms movement because you just don't know and it's hard to predict. Okay. Why do you want to do something like this? This is an example of a collaboration with Colin Cleanhouse. They wanted to know about collagen B and how it gets into the cell. So they had this transport protein and the collagen, and we predicted how it bound on the outside. And then we started pulling the N-terminus through and predicted other dock structures. Meanwhile, they did NMR or fluorescence experiments and labeled different residues, and we're able to corroborate some of these structures. So you end up getting a molecular mechanism. How does the machine work? And that's why we want to do the docking. Okay, so I want to switch to diffusion models and give you an idea how diffusion models work. For the last year or so, this is the hot model in machine learning. So if you've done DALI, this program that you say, I want a picture of a scientific seminar at a Quantitative Biosciences Institute, and it'll generate a picture for you. That's a diffusion model. And here's how it works. It's a score-based generative model is what they call it. And what it's doing is learning energy gradients. So this is a demonstrative from the web. It's a 2D picture where there's some energy potential surface, and each of these dots are randomly diffusing, and they go downhill and find the minima, right? What the diffusion model is going to do is have lots of trajectories of dots moving it around, and you're going to teach it to learn these arrows. Which way should you go to find downhill? And the way we're going to do that is by knowing what's at the bottom. At the bottom of the hill are the actual data points that you see. Real protein dock structures, those are at the bottom of the hill. And then if we add noise and mess up that dock protein structure, we go uphill, and then we would want to know which way to go downhill. Does that make sense? So same thing in the DALL-E for the art generator, you can feed it a bunch of images and say, these images are all at the bottom of a minimum. And then when you add noise to them, you add static or mess them up, they're not correct anymore, and you need to know which way to go down. So it's going to learn these arrows of how to go back toward real paintings or toward real protein structures or dock structures. Okay, so this is how it works. The probability of seeing a certain structure, if we remember our stat mech, can come out of our partition function where we have some free energy, some Boltzmann factor over a partition function. So that would be the probability of seeing one particular point at some place on this landscape. If I take the logarithm of that and the gradient of that and then do some math, I end up getting the gradient of that energy back. This is just stat mech. So the gradient of the energy, this gradient, I can get out of knowing the probabilities of the partition function. So when I say I'm learning these arrows, I'm learning the gradient. What the score-based generative model is doing is it's learning the gradient of the log of the probability. The probability of what? Of different dock structures, or the probability of different images or paintings. So what's the probability of seeing a painting is what it's not learning the probability, it's learning the gradient of the log of the probability to do that. So, and it looks a lot like energies. Okay, so the score, when they say, and this is a terrible terminology, we usually call a score in Rosetta, a score, if it scores well, that means it has a good potential, a good energy. They use the score to mean the gradient or the force, which is really confusing. So, so what? So one of the questions I was wondering, it looks like energy. Is it really energy? Is it learning the same thing that I think of as energy? If I see what it's learning, is it like Rosetta? What Rosetta would do, which we've worked on for 25 years now. Is it learning a similar thing? Okay, so here's another picture of how this works. Here's an image. You add noise by adding static. You do this with a forward differential equation. And then what you're trying to learn is how to come back. And when you reverse that differential equation, you get this one and you get that grad log P term in. And that's what it's trying to learn. And you can see how this is done, right? If you're here, it's just a little fuzzy and you just sharpen the edges. Or out here, you can just barely see the dog and you gotta fill in the head. You can see how this is done, right? Your brain, you can see where the next step is. So and you're doing this in small steps. So you give it a time between 0 and t or 0 and 1. And so you put that much noise on, and then you try to figure out, how do I come back one step? And your brain can kind of do it, so you can kind of see how an algorithm might be able to do that too by learning this direction. Okay, so here's the proteins. So for docking, one of the neat things about docking is I can make this problem really simple. So the question is, how do you add noise? Right? So in docking, the simplest way I can do it is rigid body docking, where I'm just doing translations and rotations of the second docking partner and keeping one fixed. And so that's what I'm doing here. I'm adding random rotations and translations to gradually add more and more noise. And then I want it to learn, if it pops out here, it should know that it's got to come back together and get in contact. Or if I mess up the hydrogen bonds, it should figure out I got to fix those hydrogen bonds, right? Okay, so that's what it's doing. So we put some noise in, we perturb the pose, we have a graphical neural network, an equivariant graphical neural network that treats three-dimensional things nicely, and we try to learn those gradients. And then we predict scores, which is the energy gradient, which if it messed up this way, it should go back this way. And you know the right answer because you know the one you started from, so you know the direction it should learn, what arrow it should go. And if it doesn't, then you say, oh, fix my neural network and make it do something else to try and better learn this gradient. Okay, that's not too important. The loss objective, you're trying to learn these gradients of the probability by learning those scores. Part of this is deterministic. Part of it is stochastic because you have a stochastic differential equation to noise it. When you come back, you also have noise in it. And that's part of what makes this generative is as you add in noise, if I run DALL-E 5 times asking for a seminar room of quantitative bioscientists, I'd get different pictures because it would have some noise in denoising, or if I take that dog picture and denoise it from here, I might get different things depending on the trajectory it goes with a little bit of noise. So that stochastic part is important. Okay, so here's what it looks like, kind of. Here's just translation, which is three-dimensional. Here's rotation, which is three-dimensional, and the special operation, special symmetry, three-dimensional symmetry group. And then here's translation plus rotation. And then these look beautiful, like it's diffusing back, but this is actually the forward noise, and we just reverse the movie just to make it look pretty. Okay, so does it really work? Well, to make it work, we take many docking targets and we run the noising, and we train on many structures, and then we denoise on a test set that's independent. And these are some classical docking methods. These are some new deep learning methods, and they're terrible. And then this is actually GeoDoc is ours. That's an AlphaFold-like method, and it's okay. This is success rate. And then this is the generative model, and I generate 400 samples, and I say, are any of those any good? And sometimes they're good. A lot of them, at least if I just translate things, I translate back and find good structures. So that's pretty encouraging. And again, it's super fast, seconds instead of hundreds of hours, which was what Rosetta would take. Right. Okay, so this is over all these test targets. This is the docking quality. 1 is perfect, 25% is acceptable. We're almost always making good structures, good acceptable structures. I really want to be, high quality structures are like 75% and 50%. I'm really making medium structures when I'm just translating. Of course, this problem's been solved. Rigid body docking, everybody can do if things don't move, right? But this is where we're starting. So with rotation, we get about the same if we do translation and rotation at the same time. Now there's 6 degrees of freedom. The hit rate drops considerably. So more degrees of freedom. If I start moving the backbone, this is going to get worse. And that's on our to-do list, but we haven't done it yet. Okay, so we make 400 of these. How do we know which one is correct? Well, I can throw it into Rosetta and have Rosetta calculate the energy. So this is a Rosetta energy with the translation decoys. We get this nice deep energy funnel. This is RMSD. RMSD 0 is the correct answer. So the low energy is the right structure. It's high quality. It's beautiful. With rotation, it's a little harder. I get some false positives. With rotation and translation, it's harder to sample things. Let's see. That's putting Rosetta in, but I told you the score function should be the energy. It's the gradient of the energy, right? So an undergrad, Jackie Chen, started asking, could we use the score to rank these models? The thing that the machine has learned, maybe it's better than Rosetta. So what we did is we take, as you're diffusing here, if you have grad of the log of the energy, if you integrate that as you go, integral of grad log E should be delta E, right? It's a little more complicated than that because the time variable is also changing. Because the time variable is changing, it actually looks more like fluid flow, and you need to integrate the divergence of the grad log E as you go. So it's a little more complicated, but the math holds up. This is the delta log likelihood that we get out of the trained model, the diffusion model, for 2 different cases versus the Rosetta energy. And you can see here, we have a nice energy funnel. It's learned an energy function, and the lowest energy is at the correct position. That's pretty good. This is a case where the lowest energy it thinks is 8 angstroms away, whereas Rosetta knows pretty clearly which interface is correct. So it's learned some kind of energy so far, but it's not as good as a physical energy so far, and it's not learning the exact same thing. So that's kind of where we are. There's more work to be done to break this down and figure out, maybe there are ways we can do better again by combining the physical and the machine learning approaches. Okay, so hopefully I left a little bit of time for questions. The summary, I taught you about language models and the Antibody language models for antibodies. I talked to you about the 3-dimensional prediction of antibody structures, the generative model for producing antibody sequences and libraries, and then this diffusion model to make doc structures. And hopefully I gave you a decent picture of how the methods work and how you might use these. I have to give you a shameless plug while I'm here. I run a summer intern program and a post-bac program where we train people in Rosetta methods. I send people to Rosetta labs all over the world. The applications are now open. So it'd be great if you know some talented undergrads who might benefit from a summer with us or a post-bac year to get ready for grad school, please send them our way. And then so many people in my lab, we really retooled everything. When Joey was in the lab, I could be lazy and be like, okay, it's just Rosetta, do this and that. And I had to relearn everything to do all this machine learning. And I basically learned from these students because they figured it out and then taught me. So Li-Shin Chu is doing the diffusion work. Jeff Ruffalo is doing the antibody work. And then Jeff had 3 of these interns and 3 summer interns. And then this undergrad was mentored by Li-Shin. And then Sai Pooja Mahajan was a postdoc that kind of helped hold the whole thing together and guide us through all the math of this. So thank you very much for your attention. I'm happy to take questions if you've got a few minutes.
Thank you, Jeff. That was terrific. So we've got 70 odd people online and probably more than 70 in the room, and time for a few questions. So who would like to start? Sagar?
Thank you. That was really cool, Jeff.
Thanks.
My question is about the use of AlphaFold for training IGFold.
Yeah.
And you said that, with with analogy to machine translation. So did you actually observe that if you withheld some of the AlphaFold, did the performance degrade or, and so on?
Absolutely.
So can you comment on that?
Yeah, if you just use the PDB structures and train, you get a certain level of performance and error. If you add in all those other synthetic structures, the performance is better. The RMSD predictions of held out H3 loops are better. Absolutely. Yeah, I have
how is that happening?
I know I don't, well, okay, we didn't use any old AlphaFold structures. We only took ones where the confidence was good. So I think they are better structures. And then I think it just, you're training this brain, this network to do things. It needs practice. It needs lots of practice, and you're giving it more things to practice on and more cases. So it makes it more robust. Yeah, that's the simple answer. I have the postbac in my lab in that Rosetta postbac program is kind of looking at training schemes now, and she's going to try to see a little more in depth how that affects it. And if we use IG fold structures instead, does that do better? Because now we have 1 million structures. I could use 1 million structures to train on.
Yeah. Before we continue in the room, there is I think a relevant question online in the chat.
How do we account for crystal packing? I don't. Yeah, crystal packing might be affecting things. I don't have that in there. That could be why. The limit we seem to be at now is about 2.5 angstroms resolution on the H3, and maybe that's because these loops are flexible and they're stabilized by crystal contacts. Maybe that's why all of the deep learning methods seem to be stuck there right now, or maybe not. Maybe we're still missing something about loops and we can get better if we're better at it. I'm not sure.
So a thoughtful triaging of the training set might solve that problem.
Right. Again, more data is better. So I'm reluctant to cull some, but that's a good idea. We could try that. We could take ones that have more crystal contacts and take them out and see if that changes anything. That's a good idea. And I think this postbac could do that. Yeah.
That was a really great talk. Thank you
Thank
so
you.
much. I was just curious, in one of your slides, you were comparing some things with AlphaFold 2, and one of the things that I was noticed was part of that plot. Sure. Was part of that plot, it seemed like there were some outliers in the AlphaFold 2 that your method actually came up quite good. And I was curious if you could say a little bit more about what the heck that was. I think it was on slide 32.
I think it killed my screen sharing here.
Not to have you go back. And
Yeah.
It seemed like there were 10 angstroms RMSD.
I should know this. I mean, the picture, AlphaFold's loop was over here and our loop was over there, and the crystal was over here. Yeah.
I was just curious.
Yeah. No, I haven't figured out what the simple explanation is. On the other side, AlphaFold gets it right, especially when there's a long, that case where there's a helix in the loop. My model hasn't learned enough to make helices, but that's bread and butter for AlphaFold. So those where there's longer things and different structure, secondary structure in there, AlphaFold's definitely the right one to use. But no, I don't know what it is in our model that does better than AlphaFold. I don't know. Yeah, good question. This is the crazy thing with the deep learning models. You don't know. You don't know. Yeah.
Focused on structure.
Yeah.
I like to think in terms of making predictions, it's also requiring some information about dynamics. I
Yeah.
was wondering.
Yeah, the question is about dynamics. I think dynamics are, that's a big open frontier, work to be done. And no, I don't know. I like the low free energy state. I love crystal structures. Yeah.
1 qubit from density is 1,000 downwards of NMR.
So I do think that's a worthy problem, and people in my lab are thinking about it. I'm a little nervous about taking on that problem, but I think it's definitely where attention is going to shift in the next few years. Yeah, thanks.
So, since 2019, have you learned any new physics from these models?
Nope.
No, that's,
Yeah.
I think all that physics comes from the 19th century.
Right.
Yeah.
That's
but,
right. Well, it does, but we know Coulomb's law doesn't work very well in Rosetta. It just doesn't.
Exactly.
Actually, Sagar does better with it than we do, and with the amber stuff that works
It's-
better,
it's
but it
that-
doesn't work very well in the
that damn
Rosetta.
dielectric constant.
The
Yes.
dielectric and the hydrogen bonds and all that. So
Trips you up every time.
Yeah.
So
sorry we didn't leave enough time for student questions. I'm happy to stick around if students have things to talk
students
about.
are going to have at you for an hour.
Oh,
so
good,
that's,
good. Okay, all
That's
right.
not going to be a problem.
Great.
But I did want in front of this audience to present you with this handsome RCSB PDB Yeti with a hotshot cap. Take good care of that, and your grandchildren will be able to inherit it.
Thank you.
Okay, thank you. That was a spectacular talk. The students will join me and the speaker upstairs now. Thank you.
Thank you.
Congratulations.